Individual cells must quickly repair any disruptions to their plasma membrane induced by normal wear and tear or by traumatic injury, as Susan Parkhurst from the Fred Hutchinson Cancer Research Center in Seattle explains.
When you have a tear in the membrane, what's thought to happen is the actin cytoskeleton redirects vesicles to the surface, and they fuse to each other and then to the membrane to stop things from flowing into a cell, and then you have to repair the membrane and repair the underlying cytoskeleton. But exactly how that's done wasn't known, and it's a little bit different depending on which system you're using.
Most studies of single-cell wound repair have looked at xenopusoocytes, sea urchin eggs or cultured cells. But Parkhurst and her colleagues, Maria Teresa Abreu-Blanco and Jeffrey Verboon, decided to use a different system, the multinucleate early Drosophila embryo.
It's a big cell. It's really amenable to imaging and amenable to genetics, and genetics is something that hasn't really been used in the wound healing field before. And so we would be able to find mutations that would affect different processes, and we'd be able to define more precisely what the steps were instead of having it more nebulous.
The researchers began by observing wild-type embryos as they repaired holes induced by laser ablation.
We could see that immediately after you wounded, there was always an expansion phase. So the wound gets much bigger, which was surprising to us because the embryo is not under tension at that point. But we could see that there was definitely an expansion. It would reach a maximum size and then sit there for a little bit, and then it would start to contract. And it would get to a fairly small hole, and then there would be a long phase where it finally knit together the very end part and made it right, which we call closure.
Actin was rapidly recruited to the edge of wounds and, just as in other systems, this accumulation was required for membrane repair. Blocking actin polymerization with the drug latrunculin B resulted in greater wound expansion and impaired contraction and closure. Parkhurst and colleagues then looked at the microtubule cytoskeleton.
We wanted to see if microtubules were all of a sudden changing in their pattern. We didn't see any changes, which is different than, say, the Xenopus model, where they see radial microtubules from the wound edge. We don't see that. But we thought, well, are they important? And in that case, we also used a drug inhibitor to see if we remove them, if they were required. And the answer is yes, they are required. But they're not doing exactly the same thing as they do in other systems.
The researchers then turned their attention to the motor protein myosin II, which co-localized with actin at wound edges. Blocking myosin function by either mutation or drug treatment prevented the embryos from closing surface wounds.
What we think is happening is that there's an actin-myosin ring that's formed just below the membrane, and when it contracts, that's what's helping the hole to close. So it's pulling the membrane and everything else to actually cinch the hole shut.
In other systems, intracellular vesicles fuse to form a membrane patch that quickly seals holes in the plasma membrane. In Drosophila embryos, Parkhurst and colleagues could also see vesicles form a plug beneath the actin-myosin ring, and inhibiting actin, myosin or microtubules blocked this membrane recruitment. The patch may act as a scab that covers the wound while actin and myosin cinch the hole shut. But how do actin and myosin associate with the plasma membrane?
So if it's really pulling the membrane shut, it has to be attached to the membrane somehow, and it was really interesting how that could be. And there just aren't that many proteins that are known that have the ability to link the cytoskeleton to the membrane. And so we said, well, we should test the ones that we know about and see if any of them have a role.
One protein that Abreu-Blanco et al. Looked at was the intercellular adhesion molecule cadherin.
We didn't really expect that it would be that one because cadherin is not thought to be expressed in the early embryo, and it wasn't until after that we could see that it really was, but it's at a very low level. If you didn't know it was there, it would look a lot like background until you can see that it's accumulating in response to a wound.
Cadherin was recruited from both the plasma membrane surrounding the wound and from intracellular particles that traffic toward the repair site. And cadherin accumulation was blocked when myosin II was inhibited. Importantly, actin-myosin ring assembly was disrupted and wound repair was delayed in cadherin mutant embryos, suggesting that cadherin helps coordinate both membrane and cytoskeletal dynamics during single-cell wound healing.
So we see vesicles coming from below to plug the hole, so we'll see that happen very, very rapidly within like a minute. And then you get the assembly of the actin-myosin ring right below the membrane. And then it will start to contract. And as it does, the membrane will become more solidified around the wound. And it will continue to contract until it's a very small spot. And then you get sort of remodeling of the membrane and the underlying actin cytoskeleton. And that takes a little bit longer, more like 10 minutes to 15 minutes.
So, having developed the early Drosophila embryo as a system for studying single-cell wound repair, what do Parkhurst and colleagues want to investigate next?
I mean, it's not really known what the start signals are, so how do you know you're even wounded? And one of the things that we found, actually, when we were doing some of the preliminary studies, is that it matters how you wound. So if you do mechanical wounding versus a laser, you get different responses initially. So what we want to do is to look for what molecules are involved in sensing that you're wounded and being able to sense what kind of wounding and what kind of repair that then brings up. What's happening during the expansion phase? Why do you need to expand? And when you start to contract, do you only do it through this actin cable, or are there other things involved? And when you're finally to the final steps where you're knitting it together, what exactly are you doing? And how do you know when you've finished? So all those things are possible to start filling in all the gaps when you have genetics and cell biology.
And you can read more about the process of single-cell wound repair in early Drosophila embryos in the paper by Abreu-Blanco et al., published in the May 2nd, 2011 edition of the Journal of Cell Biology.